Okay, good evening, everyone. I think that we can begin today's meeting. I hope that the rest of the participants will join us soon. And today we have the fourth meeting of the current session of our seminars. And the topic of today's meeting is genome-wide association studies. And today we'll be having two guests as the main speakers. So first, we have Nikita Artemov, who some of you may remember as a speaker in the first meeting. Oh, Nikita, hi, can you hear us?
Yes, hi everyone. It's a pleasure to be here.
Yeah, thank you for joining us. Yeah, just a reminder that Nikita is a professor at the Nationwide Children's Hospital in Columbus. And besides Nikita, we also welcome, are very glad to welcome Yakov Tsepilov. So Yakov, can you hear us?
Yes, I can hear you.
Great. Could you please say a few words about yourself as a brief introduction?
Well, I have the proper slide about myself. I'm working in Wellcome Sanger Institute in Cambridge, UK. I'm genetic analysis team lead in open target genetics. We are working with GWASs and target prioritization. But I will elaborate a bit more when will be my turn.
Okay, great. Yeah, thank you so much. Yeah, just as a reminder about the format of our meetings. So basically, we have usually the introduction part and then the discussion part. And for today, I know that we have some things to present and share from both our speakers. And then I think that we can start with an introduction from Nikita, and then probably give floor to Yakov to elaborate a little bit into what he just said, and then turn to the questions and answers part according to the set of questions which we have for today. Is this okay, Jakub, with you? So as you mentioned that you would like to show something, so is it okay if we go like give word to you? Well, if after the first introduction, but before the questions, is that fine?
Yeah, that's fine.
Great. Then let's begin. And yeah, Nikita, please, the floor is yours. Yeah.
Thank you. And if you can just enable the screen sharing in a meeting, that would
so
help
it is not
enormously.
Okay. I see. Yeah, let me check. Yeah, I think it will be okay. So here, yes, it should work fine.
I'll be quick. So in the introduction, based on the previous meeting, we had some of the, I kind of understood the structure of the whole endeavor. So I'm just gonna give like a 3-minute overview of the topic of discussion and maybe some of the things to elaborate on during the discussion part, and then I'll transfer it to Yakov later on. So basically, we're talking about the genome-wide association studies today, and this is the scenario where we will have a cohort of cases, a cohort of controls. We're interested in figuring out what's going on in their DNA and what are the differences. So basically, we'll have a genetic information, and usually that will be performed with the microarray genotype, and we'll have the information about the genotypes from cases and controls, and we'll compare the prevalence of the allele frequencies, for the specific variants. The key kind of feature of the GWAS is that we're not looking on in the coding sequence only, but we're actually capturing more of the non-coding signals. And it's actually, we're lucky to have some in a coding sequence in the GWAS because it's easy to interpret. So we're looking for a relatively short chunks of the chromosome that are associated with the disease. Basically, quite often, we not necessarily will be able to identify the specific mutations which are causing the disease in a particular individual. More likely, we'll identify just some variants which are linked with the actual biological cause of the disease. So and basically, since there's a lot of statistical considerations which we're going to talk about in this meeting as well. So the fact that we're running 1 million of such comparisons is probably we have to adjust for that. This gives rise to the genome-wide significance thresholds for these comparisons, and so on. So basically, the background and origin of the GWAS studies are actually, identification of this small chromosomal regions, which we will call haplotypes, which originate from HapMap project. So these are basically the co-inherited regions of the DNA, which are transmitted together, and we're therefore able to identify the genetic signals which are corresponding to haplotypes rather than specific variants or genes. And basically, this structure of the haplotypes is dependent on the genetic ancestry, and as a result, it's important to control for the ancestral composition of your data set or adjust for it whenever you're running the GWAS study. Importantly, what was the field was able to figure out with the HapMap project is that we're also able to create a short representation or digested representation of the common variation that is present in each subject, and this is so-called creation of the tag SNPs, where we're able to take the entire sequence of the DNA and figure out those positions which are commonly different between the individuals and are variable, and therefore reconstruct the kind of reduced representation of the entire genome. And this is why with this kind of genotyping approach, not the sequencing approach, we're still able to call this a genome-wide association study. So basically, this leads to the idea that we don't need to know the entire sequence of the genome. We need just only these tag SNPs, which will be representative of the respective haplotypes. And therefore, we can have this amazing technology with microarray genotyping, which we can use to simultaneously uncover the genetic data for many variants and many individuals at the same time at a reasonable cost, which enables the creation of the huge data sets to basically get down to the association study. To date, we have seen in the field a large amount of data being analyzed, including the meta-analysis across the multiple biobanks, so high-profile cohort with genetic and phenotypic and often longitudinal data, basically enabling the GWAS studies across many phenotypes, including the resource that has been built up by our lab and now available to work with the genetic data within Russian cohort. So it's a biobank Russian resource, which we have created and is now accepted and should be out soon in the publication. But it's available as a preprint, and it's basically a data set of about 4,500 individuals with the genetic and phenotypic data, with about 400 GWASs available for the entire cohort, with a whole summary statistics for GWAS available for download right now. And we built up this as a resource that is now available online to everyone, and basically just illustrate how different or not different may be the Russian population from the European part of Russia in the context of the UK biobank and the other large studies with replicating some of the known genetic associations, as well as boosting power for others and finding some genetic associations that are specific to Russian population. Very few, but nonetheless. So basically, I'm excited to discuss this topic for the next hour. And with this, I'll move to Yakov, to the microphone for Yakov to introduce himself and continue the discussion.
Okay, thank you, Nikita. Very nice. Well, I guess all questions are going to be after my part. Okay.
Yeah.
Very nice introduction. Very good overview about GWAS and everything. Probably the only one thing that I want to emphasize here is fine mapping part. So let me share my screen. So I will not discuss about like GWASs in general. It was very well covered by Nikita. Like, let me share the screen. Blah, blah, blah. So Nikita mentioned very important part is like GWASs, they indicate the region in the genome which is associated with the trait of interest. And it's not really so straightforward how basically you're gonna, well, I just let me show you. Yes, you can see my screen. Yeah, perfect.
Yeah.
It's not really trivial how you're going to assign exact genes that are associated with a trait and how you, well, more specifically, which variants are exactly responsible for the association, something that is called causal variance. So the method that helps to infer, at least at some level, the exact association, exact variance, causal variant is called fine mapping. This slide illustrates in very simple manner what does it mean. So originally from like a Bayesian statistic, there are methods to reduce dimensionality of the data. So basically, if you have a lot of SNPs, some of them are significantly associated, some of them are insignificant. So using several approaches, you somehow can reduce the amount of information to pinpoint variants that are with some probability are causal. And this probability to be causal is from Bayesian statistic is called posterior inclusion probability, or PIP. This matrix is pretty much common for all fine mapping methods because what is basically all fine mapping methods aim to do, they just want to convert the GWAS that usually consists millions of SNPs, like 10 millions, 20 millions, into the very restricted set of the variants that are most likely causal. The second term I need to describe is what is credible set. So on this very simple example, you can't really say which variant exactly causal because posterior inclusion probability is not really that high for any of the SNP in the region, but at least you can select top M variants with the highest posterior inclusion probabilities, with the sum of these posterior inclusion probabilities equal to the specific threshold. Usually we use 99%. So it will form something that is called credible set, that is just list of SNPs with a specific cumulative sum of PIPs. These are just an examples of how it can be in like real situation. So after performing fine mapping, you potentially can have loci that have pretty low posterior probability. So basically, probability is just spread among all SNPs in the region. Or you can have pretty much clear answer which SNP is most likely causal. For example, like top and bottom parts of the figure, like here you have SNP with PIP more than 90%. There are a lot of speculations about whether we can consider SNPs with high posterior probabilities, can we claim it as to be causal? And the answer for this question, yes, we can, but if all assumptions are met. And there are three assumptions. So the first one is true causal variant is among the genotype SNPs or imputed SNPs. Well, anyway, in the SNPs that you are using in the panel for performing fine mapping. If the causal SNP is not really among the variants you are testing, well, you can't really obtain the precise credible set. You don't have outliers because the second very big problem of fine mapping is it's kind of statistically fragile approach, and in case of heterogeneity between your linkage disequilibrium and GWASs, you potentially can have a lot of false positives. And you need to take into account secondary signals, because there are a lot of cases when you have the region, but number of causal variants is more than one. And it's kind of difficult to say in advance how many variants do you expect to see, but usually people assume no more than 10. But this is something that definitely up to discussion. And the last thing, last year in November, there was a paper when authors introduced the metric they called replication failure rate. So they downsampled UK Biobank, performed fine mapping, selected the variants with high posterior inclusion probability. Well, in their case, it was like more than 90%. And they performed the fine mapping again, but on the full sample, and see like the replication rate, how many SNPs you actually can detect if you analyze bigger sample sizes. And they detected that for 90%, the replication failure rate was approximately 15%. That's not really that bad, because, well, you do under the absence of any statistical noise, you would expect 10% replication failure rate. So they have only like 5% higher. That's still pretty much a good thing. They also introduced the method that potentially can make it replication failure rate smaller, but this is not really the topic of this discussion. So yeah, this is only one thing I wanted to share. In open target genetics, we work with a lot of GWASs in open access, and well, not only in open access, but we are trying to fine map them, and we are trying to prioritize which genes are associated with within the region using different functional omics data. Just one thing to emphasize. Yes, the short summary. Just ignore it from my previous presentation, but the only one thing. Credible set also represents one causal variant. So if your GWAS resulted in, for example, 10 credible sets, it means that you potentially expect to obtain 10 causal variants. So that's simple. So all fine mapping approaches, they aim to convert your GWASs into much smaller data set that simply consists credible sets. That's it.
Great. Yeah, thank you so much. Thank you for both Nikita and Yakov for the nice introduction. And now I think we can move to the discussion part. I've seen that there were some questions in the chat, but I think that they have already been answered. Thanks to Nikita for that. And yeah, I think we shall go basically along the analysis workflow with our discussion part. So at least will be the logic that I will try to stick to when asking the questions. So basically, yeah, as a brief reminder of what we've just heard, yeah, we have essentially the genotype and phenotype data as input. And the genotype data, which are usually generated using genotyping arrays, though I think that there is a trend towards moving to sequencing data. And basically, the result which we want to have is, as Yakov very nicely introduced, the logic and the method of fine mapping. But again, in the ideal world, we want to get not only the information about which loci are important for the trait we are studying, but ideally, we would like to have pinpoint specific causal variants which are contributing to the risk of the disease we are looking at, or to the value of some other trait which we are studying. And there are a lot of steps along the way, and we'll start with working with the actual source data and then proceed through the analysis to this interpretation part. So my first question will be, and that's basically a thing which I was personally interested in. I have to confess that a lot of my questions are driven by my personal interest in this. So basically, I think that quite some years ago, when I first came across the different GWAS studies, I think that genotype imputation in those times, at least that was my impression, that it was more of something which was important again to do the actual fine mapping, and not as a like general pre-processing step before doing the association testing. At least again, that was my impression. Maybe that was wrong, but as I can judging by what I see in the present day data repositories, for example, I see that more and more data sets come out as already imputed. So basically what genotype imputation is, again, as to those of you who are not aware of it, is a idea that we have some variants which we have genotyped in our array, and we know the genotypes for these variants, and these are usually like a fixed number of around 1 million or so. And then we can do the prediction of the variants, genotypes for variants which we did not genotype directly, but which we can again predict confidently by looking at the linkage disequilibrium structure and the haplotypes in the population, and then just by looking at the variants, the SNPs that are around the one which we want to predict, predict basically the most probable genotype. And I think that a lot of, for example, the UK Biobank dataset, the FinGen dataset, I think that all the summary statistics of the analysis which you can get are already imputed. So basically, that raises my first question regarding the imputation. So is it really something which should be done routinely before the association testing for all cases, like for all studies, or not? So what's the best way to do it? Is it like a necessary step or not that necessary step? And so if you can say a few words about that, let's probably start with Nikita.
All right. So conventionally, there are two, at least I kind of prefer to think that there are two reasons for having imputation or having, creating an idea of imputation. First, there are different types of microarrays that are being used, and they are using different variants. So if you genotype something on one microarray, somebody, your friend has genotyped this on the other micro, your data is incompatible. So basically, one reason for having imputation is to make the meta-analysis possible, because by running the imputation procedure, you're able to reconstruct, if not all, but a lot of the shared loci, and add some more on top of that, and basically create a meta-analysis for a lot of variants which will be present in both data sets. The other, so this is just a technological kind of standpoint of view on imputation. The statistical point of view on imputation is the following. By running the imputation, so your directly genotype variants create a relatively sparse representation of the variation in the human genome. And by imputing extra variants on each haplotype, what you're hoping to obtain is the variant which is in a tighter linkage disequilibrium with the actual true causal variant for a disease, which you may not even see as a part of your data. So through imputing the more variants, you're increasing the likelihood of finding the variant which is tightly linked to the causal variant, therefore obtaining the genome-wide significant association. So this is why you will naturally see that there are a lot of public solutions for imputing the data, and all of the like TopMed, HRC platform, Haplotype Reference Consortium. So there's a large data sets that could be used as the reference for imputation process for your own data that are open to public. You can just submit your data there and get the imputed data back. And the reason is because it's so fundamental to increasing the power and making it, usable by not just you, but through meta-analysis as well. So that's my take.
Okay, so basically, as far as I understand, maybe I will direct the question to Yakov now. So the question that I have is basically the idea of imputation versus fine mapping. So basically, if you have a data set which is very deeply imputed and you have very low frequency variance, that are imputed. And again, this is just a personal experience of what I've seen, for example, in FinGen, when you have like top variant in the locus, and it's almost usually like almost like in at least like 90% of cases, this is what I have seen. Usually is the one which is has the highest PEEP score in the results of fine mapping. So that's essentially makes I don't want to say that fine mapping is useless, but for me that raises the question as to what is like the proper relationship between these two steps. So basically, if you have a data set which is like well imputed to very low frequency variance, like what's the rate of new discovery, like or new variants that will pop up in fine mapping, for example. If you can maybe say something about this, so whether these steps are really like whether the deep imputation does make fine mapping less necessary or not, or is it just about getting a variant with a higher linkage to the causal one? So what's the idea?
I need to give a context to this problem because originally imputation was introduced to solve the problem with meta-analysis described by Nikita. And imputation panels that were used on like 15 years ago, their coverage wasn't really even close enough to like at least be sure that you cover all variants that are potentially can be causal. So no one really relied on imputation as a potential solution for problem of fine mapping and genetic coverage. It was solely about making panels comparable with each other and to perform meta-analysis. Just for the reference, the old HapMap2 panel for imputation, it can give you roughly 2 million or even less SNPs. Now, all panels like HRC, TopMat, and even newer, their coverage actually, well, it's just basically limited by the size of the reference, but usually, I don't really have any figures with me right now, but it claimed that it can robustly impute more than 90% of the common variation in the genome. So here we actually need to specify two different things. So common variants and rare variants. And for imputation, for now, as like a modern tool that is very, very well sophisticated and with a lot of gold standards that are presented in the market, it probably solves the problem with common variation. You're still like missing something, but you know it will be more than 90% will be covered. As soon as you go to the space of rare variation, it all becomes quite tricky. Because first of all, you can't really impute the rare variation and like the quality and the coverage is, well, obviously it reduces as soon as you go to the toward like the rare variation. But if you're lucky enough, you still can gain quite a lot of stuff, even if you perform your fine mapping, even like as is. FinGen is a very nice example. They published the paper in Nature last year. They didn't really went too much into the details, so they simply fine map their own cohort using DNA array, using like openly available references, and they enhanced it by their own sequences from their own population. And they performed fine mapping for everything toward like mineral frequency 10-4. So it's like basically like rare variation, really. And they detected quite a lot of powerful, quite a lot of significant rare missense rare variants, just like performing as is. They don't claim that they actually covered rare variation like entirely, but if you're lucky enough, you will cover like everything. You will cover at least part of whatever you can catch there. So answer for your very first question, whether we need to apply it, yes, we need, especially if you are focusing on common variation. If you're focusing on rare variation, yes, you still need to apply.
I'll only add to that. So this is like a really amazing answer. So in a way, I'll just add to that you should think of FinGen as a very unique project in terms of the microarray that was used to genotype that is a UK biobank array plus mostly plus the Finnish specific rare variants. So you can kind of get the decent coverage of the rare variation on the microarray itself. So, saying that whatever, the strategy is applicable to FinGen in terms of rare variants may not be applicable to, other biobanks necessarily, mainly because of the uniqueness of, genetic background of Finnish population, where the whole project is basically aiming to search for the population-specific rare variants on a, populational scale. So that is somewhat outstanding example, for the imputation considerations. But
yeah, thank you. That does answer the problem that I was having personally. Yeah, and like closing up with the topic of imputation, I think that there is also one pretty important question regarding the reference panels that are used for imputation, because basically, yeah, we need some very well constructed, high quality haplotypes in order to predict the missing genotypes. And the question is, if we are working with some population which is underrepresented in the like projects such as the ones that have been mentioned, or for example, like the sequencing projects like 1,000 genomes or something. Yeah, is this a big problem? For example, if we're working with a population of European ancestry, but again, which does not have a very well matched reference genotype panel. So does it introduce a significant bias? And well, basically, if this is the case, whether the state should be omitted, because again, as far as I understand it, the idea here is that if the imputation, if the imputed genotypes are of low quality, they will introduce more noise than give some like additional findings. So first, is that true? And second, so like, is there really a problem of significant bias for the underrepresented populations?
Well, I can start with the answer. Probably my experience is a bit different with Nikita's. First of all, if your population doesn't match the reference panel, obviously you will introduce the bias. But it's kind of difficult to assess this bias because the imputation quality, well, you have different metrics of quality after the imputation, so you can directly assess it. So if the imputation quality is still high, even using like inappropriate reference, you probably can be safe using this variant. But what you will detect, obviously, is that number of SNPs you can consider as reliably imputed will be much less than you usually expect in the situation when you use the proper imputation panel. Of course, before you run the imputation, you will run this very simple population genetics analysis like principal components. So you will like have, for example, 1,000 genome, you will plot your population into it and see like where it lies. So whether it actually overlaps these populations you have in your reference, or at least it aligns with the major populations. But in the very end, you simply will run it with whatever you have in your hands. So we had this experience with our own cohort. We recruited it in Novosibirsk. It had only like 500 people. We imputed them, or we genotyped them using one of the DNA arrays from Illumina, pretty much common one, and then we just tried to impute them using different references: 1,000 genome, HRC, TopMed, and with different combinations of ancestries. So the only one metrics we used is like how many SNPs we actually obtain with a specific quality metrics we consider as a high quality SNPs. So that's it. And the last thing, we actually kind of tried to validate the results. So we applied, so it's quite, if you have phenotypes, and you obtain the imputation results, the first thing you need to do is basically to reconstruct PCA plots, because if you have like shift in your PCA plot of imputation, it simply means that something was wrong with your approach. The second thing, you can apply, you can calculate polygenic risk scores on your population and compare with phenotypes. For example, height or weight. If you have the significant correlation between polygenic risk scores and the original phenotypes, it means that you probably imputed it correctly. So it kind of simple tricks that can help you to control the bias.
I'll just add to that briefly that there is potentially one more way to validate. So the issue with the underrepresented genetic cohorts is that they lack the whole genome sequencing data, which can be either used in the reference panel or as a validation for your imputation. Because ideally, imputed variance should be, close in allele frequency to what you're expecting to see in the unbiased whole genome sequencing. So usually you would not have that data. So one kind of, statistical way to validate your imputation quality as well is to run the masking experiment, where you can just, mask the variance from, 10,000 variants on each chromosome, those that were directly genotyped on the microarray, so you're confident about the genotype, then try to impute them and see how, concordant the imputed and the direct genotypes will be. It's important to keep in mind that what kind of metrics will be used to assess the concordance, because usually the R-square value and so on can be biased with a relatively rare variant, have like ton of reference genotypes in imputed data, tons of reference genotypes in the directly genotype data. It's gonna have good concordance, no matter what's the imputed genotypes are. So there are several extra metrics which are accounting for the allele frequency and so on, such as IQS scores and so on, imputation quality score, basically. So we ran this experiment on the small subset of Russian data from St. Petersburg. Kind of indicates that well, it's not an isolated population, and it's pretty much will be well explained by the broad European haplotypes. So the trick is that if your population that you're trying to study is not isolated, is not absolutely absent from the whatever has been genotyped or sequenced so far, then you probably will have, with all the sophisticated imputation software, you'll probably will have very good chances of imputing a decent amount of variance with good quality. And the way to check that through this masking process, it's only the case, as Jakub correctly pointed out, the allele frequency will play a large role in this. If you're imputing common variants, you probably will be just fine. Rare variants, that there you need the dedicated specific reference population panel, which has been done in FinGen, for example. So the rare variants are imputed through the whole genome sequencing of large number of fins, because this is where the population structure will matter a lot, and you won't be able to get away with just using a TopMed or large diverse platforms. They may not have the power for the rare variants, and we saw the drop of quality of imputation for the Russian population, for example, in rare variants, irrespective of the platform, the size or the ancestry composition of the reference platform. So basically, common variants you most likely going to be fine no matter what. The rare variants you need the population specific panels. That's my take on that.
Yeah, very nice idea with the masking. Yeah, it's really good. Great. Yeah. Maybe as a segue to the next segment, I will ask a question which is sort of in between the two. So basically, the final thing which I again personally interested in. So if I have well, let's consider like the example if I have not the genotyping array data, but some sort of sequencing data set which has not a particularly good quality, let's say, yeah, and I have a lot of missing genotypes in there. And again, I use this question as sort of segue to the quality control topic. But in case again, I have several set of samples which I think that are pretty bad in terms of, or at least somewhat bad in terms of the call rate, the proportion of genotypes that are called. Is it a good idea to run imputation as sort of the fix the bad sequencing data set to add the genotypes like for the variants that have not been properly called? Or is it a bad idea and well basically you should remove the samples with low call rate altogether?
I'd say you would look at the what was your targeted sequence coverage. So if you were planning to have 30x coverage, you obtain the sample with 2x coverage in the end. I wouldn't try to impute anything in there. I would just eliminate it because there's something fundamentally wrong in the experiment, or at least I wouldn't try to publish that without validating the approach first. However, if you're thinking about the things like low coverage whole genome sequencing, which is traditionally used now as the the alternative approach to microarray genotyping, this is exactly the situation you're describing. So I aimed for like 2x coverage. I've got my sparse 2x coverage, somewhere greater, somewhere smaller in some parts of the genome. And that is sufficient to run the imputation in it. And that is basically, as an outcome, it works, at least as good as the microarray genotyping plus imputation, sometimes even better. So because with microarray, you're pre-specified to a specific genetic locations. With whole low coverage whole genome sequencing, you're basically, getting much broader coverage of the genome. So from the quality standpoint, it looks like 2x, 3x data is more than sufficient, even lower coverages people were able to use in GWASs. So basically, depends on your targeted coverage. If you're planned for it, run the imputation. If you haven't, then check your experiment.
So basically, yeah, if you have a data set of like which is aimed at 30x and you have a subset of samples which is like maybe not 2 but like 15x and a very low, a very high rate of missingness, it's better to remove them than to try to fix them.
I mean, because you will need to check how well you fixed it, and you're unsure what is the cause of that problem. So it kind of opens up a lot of possibilities for the false positive associations. I'm not sure how to control for that explicitly so that any reviewer will buy it. So
I wanted just to add one thing. We never worked with low coverage, low sequencing with low coverage and imputation. We always work only with DNA arrays. I'm pretty sure that like low coverage sequencing, they have their own rules of like quality controls and everything, because when you run the imputation on DNA arrays, you actually have to be very careful with whatever you source as input. Like there are like huge tutorials and a lot of rules and pipelines how you need to perform your quality control, because it's basically very strict. So it's much better to source less information to imputation, but be sure in the outputs and source more data, but noisier. I think the same rules are applied for low coverage sequencing.
Yeah, thank you. Yeah, that regarding the low coverage, yeah, that's a pity that we don't have. I think here with us people who are like have more of an experience with this kind of data. But generally, yeah, it's at least as far as I can see, at least it was sort of a hot topic. I'm not sure about now because it now seems to me that more and more people are moving to just conventional relatively high coverage data sets. They have resources, obviously. But yeah, returning back to the topic of quality control, which I sort of wanted to begin with this question regarding is imputation a good way to fix bad samples? So basically, the thing with the quality control, and again, just as a brief reminder to those in the audience who are less familiar with the quality control in GWAS, yeah, the idea usually is to remove both bad variants with bad genotyping rate and bad quality metrics, and also remove the samples that have also bad quality metrics and bad genotyping rate, for example. And there is, I think, quite a lot of importance, yeah, in how we do this quality control, because a lot of false positives, a lot of false positive associations are clearly resulting from bad quality control. I remember I had reviewed the paper like several years ago where people claimed to identify. They studied some sort of a rare condition which has very low frequency. They had like 100 patients, and they've claimed to run a common variant association analysis and finding like 60 loci, which were basically, the idea was that we have a 60% frequency in cases and 0 frequency in controls. And basically that was, again, the authors claimed that it was a legitimate result, which obviously I also didn't buy it. But well, those kinds of things, and we've seen them, the similar things ourselves in our own work. Yeah, the bad quality control may result in very unexpected sort of findings. And hence, so my first question will be in this topic is that, so is the set of thresholds which are like applied for this quality control step, is it fixed? So do you usually like have a single set of thresholds which are applied, or do you really look at the data to sort of see how the different metrics are distributed to find the good cutoffs? So is that really like a strict like set of like use this like 0.95 for missingness, 0.001 for like Hardy-Weinberg equilibrium, etc., or is it more sophisticated than that? Nikita, please start with this.
I mean, first of all, it is critical to not introduce any bias with your quality filtration, meaning that whatever you're doing with cases and controls should be not necessarily the same, but consistent. So, for example, if you're having a large imbalance between the number of cases and controls, so like 10 cases and 10,000 controls, the quality filtration for call rate of 10% does not make sense, right? So you're filtering out one individual out of cases, and 1,000 individuals or so, or 100 or whatever individuals out of the control. So you have to make them consistent. It's ideal when your case and control cohort sizes are comparable, then you can make it absolutely identical. But generally, I'd say that we have a good way of controlling for systematic inflation in genetic studies, and that is probably bridges to the next part of the discussion. But we do have the way to, run the quantile plots and estimate the genomic inflation, and this is usually a good indicator for presence of any systematic errors. But on my side, I definitely would be very careful with running any joint QC, whether the cases and controls are a part of the same QC procedure, because your filters may eliminate different proportions of cases and controls, naturally, creating the bias in allele frequencies. Other than that, I'd say that that's, is usually an iterative process where you run your, QC, check the QQ plot, you make sure that it's all right. You go back if some things need to be fixed. But other than that, there is a good standard, set of thresholds for quality filtration, and this is a good starting point.
It still probably depends whether your focus is common variation or rare variation, because if it's common variation, you basically don't expect to see any huge difference between cases and controls. And it is, well, basically for common variation, you supposed to quality control and impute everything together because it's usually probably one cohort. Because again, it's, to my opinion, it's kind of dangerous to perform quality control separately in cases and controls. Usually even like you do expect some differences in some variances between cases and controls, but your Hardy-Weinberg will most probably not detect it. And in human genetics, it's common to actually apply Hardy-Weinberg as one of the steps for quality controls. So answering the original question, I would say if your focus is common variation and you're using DNA array and it's human genetics, there are very strict rules, very fixed thresholds that usually you're supposed to apply for quality control. So you usually don't really apply any extra steps to infer the thresholds. The problem is when your design is not really that common. For example, well, rare variation, and it's probably very well covered by Nikita, but in our group, we worked with plant genetics and with animal genetics. And this is a huge problem there because, first of all, you don't really expect Hardy-Weinberg in populations you study. Usually, just because of the nature of how DNA arrays are produced, the quality of genotyping is much less than in human genetics. And because of the like genetic heterogeneity between animals is much higher, so the chip you designed for this specific species of animal, it's usually doesn't really work very well between different breeds. So it's a common problem there. And in case of these very specific designs, you actually have to make extra steps just to understand which metrics of quality are adequate for your design. But you need to be, again, very careful there. So I would say for human genetics, since it's like a very well-established field, a lot of studies there, the genotyping technology very well calibrated and such, there are very strict rules how it's supposed to be applied. For everything else, well, you need to be very careful.
Yeah, actually, like when I formulated the question, yeah, I was thinking of our personal experience of which we've had with, and we mostly have. We didn't have like in my personal experience, I've never worked with the like really good quality array data set, and we've tried to do some sort of association analysis with exome sequencing data, which and there you have I think more sequencing specific things and in terms of what you get, some varying colon artifacts in some cases, some variants which are not that well genotyped, and I think that in some cases that at least we've seen in our returning back to the idea of quality control cases and control sample, we've seen such certain things like variants that are despite like the fact that we've genotyped everything in one run, and essentially we have all the same settings, still were some cases where due to some like batch effects in terms of like sequencing, you have certain variants that pass some natural control, for example in GADK, and then they still have like really biased distribution in terms of that in some samples you see these variants, and you can correlate basically the presence of these variants to certain technical covariates. So and that's like just as a short follow-up to the question of like separate quality filtering. So in this case, so is it like a good idea, for example, to normalize and to check if you know some technical covariates, so for example like the sequencing run, etc. Is it a good idea to use this variable to sort of make sure that the variant calls, the genotypes are consistent in terms of like genotyping rates, for example, in these batches. So should I for example do it on before I do the association analysis, or should I do these technical covariates, like including them into the model when I already running the association test as a covariate variable in the model? So what's the good way to deal with these sorts of like essential batch effects in the data?
Well, if you if you look at the examples of FinGen, which has already been mentioned, this is more or less typical for large data sets to have more than, well, the data is generated in rounds. It's barely anyone who can afford genotyping, 500,000 of individuals in a single batch or whatnot. So naturally, you will have unavoidable batch effects. Therefore, they're usually, well, first of all, you should see the consistency between batches. If your batch is just outstanding out of all, no matter how hard you're going to try to correct that for the batch effect, you'll still see some of the footprint of that. But even in the more or less consistent allele frequencies data across the batches, FinGen still, for example, in their paper reports how they're adjusting for the variant of the release of the microarray, the version of the microarray, the batch in which the samples were collected and genotyped. So aside of the common covariates like sex and the principal components that are used in the association models, like 5 or 7 different other covariates which are attributed purely to the way how data was generated. So in that way, it's kind of both steps are important to check the consistency between the batches and not seeing anything critical in there, and still adjusting for it, because for the large data sets, you're actually well powered to pick up even relatively tiny differences between the batches. So you kind of want to account for that.
I would add only probably more example on UK Biobank. It's also quite nice example about different batches. In the first original, like the first batch, when they performed imputation and genotyping of, I think, 100,000 individuals, they actually had different arrays for the sub-cohort of people with different metabolic diseases. So it's actually like, it was like a relatively healthy cohort, but within this relatively healthy cohort, they had like people with different cardiovascular and metabolic diseases, and they genotyped them using different array. And well, they obviously introduced quite a huge batch effect, but for now, it's like, it's obligatory to include these different types of batches into the analysis. So basically, when you run GWAS on UK Biobank, you have like huge list of covariates that you need to include into analysis, like at least 3 batches, but basically you can include covariate for batch from every medical center where people were recruited. You also include principal components from kinship metrics, your study-specific covariates, and that's it. So basically, list is huge, like more than 40. Of course, having sample size of UK Biobank or FinGen, it's basically not really a big deal. You can include, well, hundreds of covariates and still your analysis will be very well powered. Again, we are talking about common variation, not rare variation. But if your sample size is like 200, well, you can't include 40 covariates into analysis. So you have to be very careful, because if you have only 2 batches and, well, several hundred individuals, you're probably fine. If you have like still 300 individuals and 10 batches, well, probably you have problems there.
Okay, yeah, thank you for these comments. Yeah, that's really interesting. And I think that, well, as we've touched already on the problem of covariates included in the model, yeah, that naturally brings us to the topic of the actual association analysis. So basically, yeah, when we are talking about association analysis, naturally, I think that in the present day and age, almost all of these studies are done with some sort of model-based analysis, not the simpler sort of contingency table-like testing. So that's usually we're talking about either like typical linear regression model or something more complex, such as the mixed models. And that's actually one question which I wanted to ask. So if in terms of like if I have my data and I've done the quality control and the rest of the things, and I want to do the association analysis. So in what cases should I prefer? So I consider them. Correct me if I'm wrong, but I think that as far as I understand, the linear regression framework is sort of a standard thing. Yeah, and then there is the mixed model framework, which I really don't know like what are the factors that should motivate using the mixed model instead of the linear model. So I assume that one of the such factors is the structure of the population, because as far as I understand, the logic behind the mixed models, which include kinship as a variable in there, they should probably do a better adjustment for the population structure. But again, I don't know much about that. So what's your take on this? Well, basically, where should I go for some mixed models and use the kinship matrix as one of the variables in there, or just taking typical linear regression and adding like PCs as covariates? So is there any like idea of where and when should I use the LMM instead of linear models?
Probably I can start. So for now, if you have huge sample sizes, you need to include both. So it's kind of not really a big deal, because again, including kinship matrix as random effect is still considered as one of the covariates. And it's like, it's one of the degrees of freedom. So pretty heavy degrees of freedom, but anyway, people usually include everything. So mixed models and principal components. It sounds a bit of overkill, because originally, like, statistically speaking, they suppose, like, if you run random effects or if you run fixed effects with PCA, they supposed to result in a kind of similar output. That's sometimes not really the case. But again, having huge sample sizes, it kind of, for the huge sample sizes, it doesn't matter. Just include everything. The problem is when you have very small sample sizes, and we had a similar cases with animal genetics where we had like sample size of 80 animals. It's like, it's small, it's tiny, and you need to be very careful what you are actually going to include into the analysis. So we spent quite a lot of efforts to prove that basically including only kinship but without principal components leads to exactly the same results. So relatively the same results. So we could just simply exclude principal components to save the power, because again, including each covariate into analysis potentially can reduce the power. So you need to be very careful here. It's the same with the batch effect.
And I'll just second to what Yakov said in terms of one of the key parts there is the amount of analysis that you're planning to do, because linear model is a standard because it is cheap. So if I want to run the all phenotypes, GWAS for all phenotypes on the UK Biobank scale, it kind of makes sense to run the linear model. Otherwise, like if I'm trying to run even the logistic model in the case control setting, that's going to be like so much more expensive if I'm going to more complex scenarios. So it's just a matter of computational cost for large data sets. So in that way, linear mixed models are thought to have produce greater power through basically enabling the analysis of the related individuals who, naturally by keeping them, you're increasing your sample size and your statistical power, but at a possible trade-offs. So in that way, I don't think there is a necessarily a correct answer here. So we can have the, linear model analysis as the first path, and then follow up on the, most promising phenotypes. Some of them will be just underpowered, no matter what tool we're using. So from a certain data size, it becomes a problem of, optimizing the computational costs and running time. So. No, Yuri, if you're saying something, you're muted.
Oh yeah, thank you. Yeah, I was saying something. Yeah, moving on from the general like testing to the actual interpretation of results, and but not necessarily interpretation, but just checking if whether the data makes sense and the quality control of the results. So I think that both of you have previously mentioned the QQ plots, which are sort of a very important diagnostic tool to look at the association results and see whether the p-values, like this summary statistics of association, are distributed, like whether there is some inflation, meaning that there are some systematic deviations from the expectation or not. And in terms of this, the analysis of the QQ plots, I think one question which quite frequently pops up in studies which are underpowered, which have low sample sizes, is whether or not we could look into the variance and the loci that have p-values which are below the genome-wide significance threshold, so like the honest Bonferroni, like 0.05 divided by the number of variants, and if we can look below this threshold, but making sure that the QQ plot is basically like showing that there is some sort of deviation from the nil, and it doesn't look like overinflated, but still that there is some evidence for the association signal. So is it actually a good idea to look into these loci and try to like interpret them and maybe use them for some sort of downstream validation, or is it not a good idea? So what's a good approach here? And if you know, for example, that you have a low sample size and you know that you are underpowered, but still you like, do you have any chance of like identifying something good there or not?
I mean, to a certain extent, you'll have a lot of troubles publishing anything which is not reaching the genome-wide significance threshold. So even if it perfectly makes sense, as a biological hit or something, it still is going to create a lot of challenge in a publication process. So therefore, you should make every attempt to actually find out, collaborators, other cohorts which may be useful to, potentially through the meta-analysis reaching that magic threshold. And even after that, you'll still need to present some kind of replication analysis which will confirm that. So basically, you have something which you strongly believe is the correct association and is not, reaching the genome-wide significance. You'll need to present extensive functional evidence on the role of this variant and association, and not necessarily the biological mechanism, but the functional proof that this is going to work. and there you're putting at risk your resources that you invest in the experimental follow-up based on the non-genome-wide significant association. So in that way, it's either you're risking your resources or you'll have very big trouble putting this through peer review process. So that's my point on that. And then I have to apologize. I'll have to drop off a little early due to conflicting meeting, but that's probably the last question I'm going to take. But thank you so much for organizing the whole thing. And Yakov, it's a pleasure to meet you as well.
Pleasure to meet you too.
Yeah, thank you for joining us. Yeah, then I think we can continue the discussion if Yakov is the the main speaker. Yeah, thank you for the response regarding the sub-threshold associations. And yeah,
I just wanted to add one thing
yeah,
about
sure.
because we had similar problem in animal genetics, like you perform GWAS, you obtain no significant results. What to do? I saw quite a lot of examples in animal genetics when people just they simply lowered the threshold of association. They saying, okay, let's just simply use something suboptimal, and this is a definitely thing that you need to avoid by any cost. So it's like don't do it any. So it's better to do something else, but never claim your results significant if you don't reach at least study level significant approach. And yes, it's kind of a big problem what to do when you don't have GWAS significant results. No perfect solution for this problem.
Maybe following up on this, regarding the lowering the threshold, yeah, that's sort of one of the painful topics, especially in some places where people really like doing this sort of massage to the data in order to get something out of it. I have seen in some cases people doing some sort of, which I've never really seen in the framework of GWAS, and I think that there is some statistical ground for that. But still, the thing is that the conventional approach to genome-wide significance threshold is naturally the Bonferroni, basically like 0.05 divided by the number of variants. But some people do lower the threshold in a way by applying some sort of like FDR correction, like Benjamin Hogberg, for example, which I think is not something which is commonly applied in GWAS. So if you can say maybe a few words. So for example, like the question which is very commonly asked is why Bonferroni in GWAS, and is there any place for any other kind of multiple testing correction?
Well, first of all, FDR is still fine. People apply it in GWAS. In human genetics, historically, people started to use Bonferroni because there was a very old paper when using permutations. I forgot the name of authors, but they somehow estimated that effective number of independent tests is approximately 1 billion. So this is why, without regards of how many SNPs you analyze in the genome. So it's like the GWAS significant threshold was set like 5% divided by 1 million, so 5 10 minus 8. But it was calculated only for European population and using the reference panels on that time. So then almost 10 years later, people recalculated it and suggested to use actually, it's not 1 million, but maybe 5 million. So it's better probably to use 10 minus 8. But still, FDR is kind of applicable even in human genetics. In animal and plant genetics, for sure. Like people apply it and it's fine.
Yeah, that's an interesting point. Yeah, because I thought that really that the Bonferroni necessity for Bonferroni stems from this idea that you have basically the same data set that you do shuffle basically in different ways. But yeah, I'm not that much of an expert in terms of whether that really necessitates a different approach to multiple testing. Yeah. Okay. So then, well, let's assume that we have identified our genome-wide significant hits. Let's assume that we have a good scenario where we have a decent power in the study, and we have multiple loci that we have identified in our association, and then we want to make something out of it. And actually, at least my understanding of the topic is that well, you can basically try to do it in sort of the statistical fine mapping way when you do identify the specific causal variants and specific loci, and then try to design some sort of like functional assays, etc., to look into what these variants actually do. But there are other methods which are sometimes applied, which are doing all sorts of functional annotation of the results in terms of, for example, pathway analysis. So there is the gene set enrichment analysis problem, something which we have personally worked with a little bit for GWAS studies, which actually has this problem of linkage disequilibrium that introduces some bias to these analysis. But my question is, so whether or not you consider these sort of like functional annotation something which is like worth doing in a like typical study. So if you have a summary statistics, then run them for all sorts of stuff to look like the expression, the gene expression data, the pathways, etc., or not. So what's basically your maybe set of things that you do for with the GWAS results? So do you just proceed with fine mapping and then look at the locus by locus, like individual basis, or do you do some sort of summary calculations with the results?
It's definitely not a short question, so it's like the topic for the separate lecture.
Indeed.
Well, I'm working on open target genetics, and our objective is to perform this something that is called in silico follow-up GWAS analysis. So it's like when you perform the GWAS and you need to extract as much information as possible from it using any information that is available. So there are at least two different ways. So the first one is there are approaches that can be applied on GWAS is like in general. And the second branch of methods, like it's approaches that you apply for loci you specify. So starting from fine mapping and localization, and then going to gene prioritization. Gene set enrichment and everything is something that possible to apply to the GWAS itself. So it's like there are a lot of methods there. So it's not like really something very fixed. For example, people like very old method that is called magma when people calculate gene-based analysis. So even without having genome-wide significant, you calculate the specific number for each gene using the GWAS results. And then having like these results for 20,000 genes, you estimate gene like the enrichment on different gene sets or tissue specificity or whatever you want. Also, like you can apply very famous and very trending approach. Well, it's kind of old one already. Genetic correlations. So if your sample size is good enough, because it will allow you to compare your GWAS with other GWASs, maybe for the same trait or maybe for similar traits. And the third one, you can apply Mendelian randomization. Again, it's not on locus level, it's on GWAS level. You can play with polygenic risk scores. So quite a lot of stuff you can do. So it really depends on the task, because some people, they're interested in like fundamental genetics in like very specific questions about the trait. Some people, they are more interested in genes. So you basically need to go on locus level. But I don't know how to what is the short answer for your question?
Well, I believe that there is none. Yeah, but the thing which I personally was most interested in is actually the idea of these GWAS level things. Yeah, because as far as at least I have seen the GWAS, the large scale GWAS studies. I may be mistaken, but I think that people not that frequently do, for all of its pathway analysis and like tissue specificity analysis, which can be done by Magma. Yeah, that for me the question was whether or not really it is something which is let's say of importance in the typical framework, or is it some sort of like approach which is not very informative and very commonly used. So whether or not people do extract some information, I understand all the idea about genetic correlations and Mendelian randomization. They're a little bit different. They are not maybe that in terms of they are not like functional genomic methods, yeah, in the sense that they compare the traits rather than look at the like properties of genes. And this pathway analysis thing is about the properties of genes, yeah. But as far as I understand, yeah, you see that the analysis with Magma is and other methods which are like that, like Pascal and others that do sort of gene score analysis and pathway analysis. So that's something which you personally think is relevant for cases when you are looking into the actual trait and trying to understand its biology. Am I correct?
Yes, and I would say that people are actually applying these methods quite often, because usually if you are focused on very specific trait, well, you just simply run everything. So you have one GWAS and you just apply whatever you have, and you put it into the paper and you publish it. So this is how people usually do it. The main problem is that whether you actually need to apply it, because, well, if you are focused on one very specific trait, maybe disease, if it's a very like common disease, most probably people already worked on it, and probably someone already like published the tissue enrichment and gene set enrichment results on this GWAS. So it's like, for you, performing this analysis in this case is not really something about the new knowledge about the data, because people already know like all these gene sets and pathways, but maybe some kind of self-validation, that basically some like proof of principle that you know pathways you are expect to obtain, and you just obtain them, and that's it. Some kind of a quality control. There is no restrictions here, but usually people do apply like everything they can, because as many information you have, as more robust your results are going to be.
That's a nice point about the validation. Yeah, it appears to me that I've never thought about this pathway analysis thing quite that way. That it can also be a way to validate something. Yeah, I maybe will have just another follow up regarding the things that you've mentioned. You mentioned genetic correlation and Mendelian randomization. And yeah, again, I will maybe say a few words about it. So the idea here, yeah, is that genetic correlations are basically just that, correlations, that they don't imply any sort of causality. But with Mendelian randomization, the idea is that you're looking for the causal interactions between the traits usually. And then the thing is that I've seen in some studies, for example, the analysis of COVID, which was published by the COVID-19 Host Genetics Consortium, they have applied both genetic correlation and Mendelian randomization with a whole lot of traits, and then try to make sense out of it and interpret it. Yeah, so maybe if you can say a few words about this, the idea of like genetic correlation versus Mendelian randomization. So do you see these two things as basically the complementary things, that really the MR is more about causality and genetic correlations are just showing that there is some sort of shared genetic components in there? are they actually like maybe more similar? And is there really? So I think maybe I will clarify a little bit my question. So the idea that MR, which I was a little bit, which was a little bit unclear to me, is whether or not you can really confidently claim that if you have a significant MR result, this is like confidently shows you that this is causality which cannot be explained away by some sort of complex network of correlations, which are just structured in a certain way. So if you can maybe just say a few words about the distinction of RG versus MR, and whether or not like the MR significant MR results really imply that there is causality, and this is not, it cannot be like explained by other factors.
First of all, genetic correlations. Well, modern methods, they're quite robust. So basically, genetic correlation probably is the cheapest thing you can estimate if your sample size are big enough. Very reliable source of information, and it's purely about like genetic overlap between traits. So you should consider this information only in the light of genetic overlap between like whatever you are studying. It's not about causal things. Mendelian randomization, yes. So answer for your question, if you have the MR signal, you can consider like some directionality between the effects, but you need to be sure that your MR was conducted according to all specifications and limitations that this method has, because MR is very, again, it's the same story with spine mapping. MR is very difficult to perform controlling all assumptions it has, and it has quite a few. And one of them, something that people can't really control like at all, is horizontal pleiotropy and absence and confounders. So if you have genetic correlation, it most probably means that any instrumental variable you select for Mendelian randomization, it has much higher. It has a much higher risk to be horizontal pleiotropic within exposure and outcome. So most probably high genetic correlation means that it's better not to apply MR like at all, because you will not be able to control these assumptions like at all. It's very difficult to properly perform MR between two complex traits. For example, you have two diseases and you want to like estimate causal signal. It's very difficult. So it's like practically you can run it, like you select instrumental variables, you run MR, but you will spend months just trying to prove that your instrumental variables are selected according to the assumptions of MR. It's much easier if you like trying to compare functional omics, for example, metabolites or proteins with your complex disease. It's much easier to control these assumptions there because number of instrumental variables is much smaller, signals are stronger, and well, you basically can simply select instrumental variable based on pathways and everything like this. So this is why MR is probably more useful when you try to combine functional omics with complex traits. But yeah, complex trait versus complex trait is still possible, but very careful.
Okay. Yeah, so I think that I have only one question which is left on my program for today's seminar, and this like sort of we circle back to your introductory part when you were talking about defined mapping, and maybe you have already like mentioned some things, but still I would like to ask this question again. If you have, let's say, a summary statistic file. Well, that's something which we have tried to do in our practice, and I think that it was not it was not a big success. Yeah, they have a summary statistic of US, let's say from the UK Biobank, and or for the UK Biobank, naturally you can get the source data. But if you have some summary statistics from a cohort that you do not have an access to, you do not have access to genotypes, So is it worth like at least trying to do fine mapping with unmatched reference panel with some sort of population panel that can be used for imputation? Is it okay to do fine mapping with us or not? So or do you have to do this only if you have the genotypes that are there that are available?
Well, this is a very dark area of fine mapping, and it's exactly what my team is doing in open target genetics. So we have like dozens of thousands of GWASs, and we are trying to fine map them using external LD. We use GNOME-wide LD matrices, GNOME-wide version 2. You have to be very careful there again because of the heterogeneity. If your LD, like the population that was used to calculate LD, is not perfectly the same as the population you use for your GWAS, you potentially have heterogeneity problem and higher false positive rate. There are methods that help you to control it. So, for example, karma outlier detection. So you can apply karma just to filter out SNPs that are obvious outliers and then perform fine mapping using new methods. But you still need to be very careful about LD. So very simple rule, your LD should be calculated based on the reference panels with big enough sample size. Not less than 10,000 people. Like 1,000 genome is not a good source of using LD. So don't use 1,000 genome for fine mapping. So this is like the obvious thing. Like if you desperately need to fine map, use GNOME-wide matrices. Especially if you have Europeans in your GWAS, well, just use European GNOME-wide matrices. They are fine.
Okay, yeah, that actually I think was the reason why we didn't succeed in our effort, because we've tried this with 1,000 genomes. Yeah,
Don't use 1,000 genome for fine mapping. It's like there are papers that actually like has the title "Don't use 1,000 genome for fine mapping."
Okay, yeah, yes, it's a pity we didn't find them at the time. Yeah, that could explain it. Why we failed bitterly. Okay, great. Yeah, that's basically all for my questions for today's topic, and it was really a pleasure to hear from you and from Nikita. Thanks to him, and unfortunately he can't hear me saying that. Yeah, that was really informative, and yeah, thank you for joining us and sharing your experience. If you have anything to add, if you have something else to say, then yeah, please feel free to do it.
Well, thank you very much for inviting.
Okay, yeah, then I think we can conclude today's meeting. Thanks to everyone who has joined and who has stayed up until the end of the seminar. There are not a few people, but still, yeah, thank you for staying with us. Yeah, and I'll remind you the next meeting will be in 2 weeks, and it will be about ancient DNA analysis and demographic modeling. Yeah, I think that we have some questions in the chat. Okay, so basically that's more of I think the questions have been answered already. Okay, yeah, then thank you and goodbye.
Thank you very much and goodbye.